But the myth that people don't know, that people always say that um, you don't put a microplate reader beside centrifuge.
Hello, and welcome to the FCBIOS podcast series, where we strive to provide the growth in education. Yeah, so that's our message for today. At this point, we have been doing this podcast for quite some time ago, but we wanted the podcast to be like more casual. So that's why we came up with this kind of formatting and to make sure that our listeners are more engaged when listening to this educational content. So, let us start with, before I start with anything, let me start with the introduction of our company, FCBIOS. So, as a company that is founded in 1989, we have like almost 30 plus years in helping and assisting customers in their daily problems in laboratories. So, we are committed to providing the best support and solutions to all customers. At this point, we have 35 plus world-class brands. We have 20K plus of customer satisfaction, and we are, well, the leading supplier and the solutions provider in the laboratory itself. Okay, so joining with me today is our, well, panelist who is an experienced technical support uh, comrade of mine, colleague, I would say. Comrade and colleague, yeah. So please welcome Dr. Zahra. Okay, maybe you can introduce a bit about yourself.
Hello, everybody. So, thank you for those who are actually clicking on this, like listening on us. So, I'm Zahra. So, I am the application specialist in FCBIOS. So, I'm dealing with pre sales, post sales, and everything related to technical stuff. Yes, you can give me a call
That's a very
during
concise.
office hours only.
Yes, that's very important, folks. Please make sure to inform all of your colleagues or yourself that every single call must be made within office hours. Okay, well, if you can see from the TV screen, our topic is microplate reader, just a reader. Yep. So we're gonna talk about microplate reader today. Yeah, you can see from there, Sarah.
Oh, yeah,
Yeah,
there's
it's
the
there.
title there. Okay.
Yep. Okay, a little known fact about microplate reader before we get to all the gist of everything about microplate readers. Actually, the history of microplate readers started back then in 1950s. Do you know about that?
Nope.
Yep. It's actually invented by this one person named Dr. Gyula Takatsi. So there was an outbreak of influenza at that time and they wanted a solution for a batch blood test. So that's how they came up with something that resembles a microplate reader at that time. He actually made like a 6 row of 12 wells. So if you count, it will be like around 72 wells. So yep, that was the first prototype of the microplate reader, ladies and gentlemen. So after that, the microplate reader was developed into a more proper structure, as what we've seen here today, into a 96 well plate. The first 96 well plate was established in 1962. So yeah, quite a history, huh, Sarah?
Yeah, interesting. It's been there since 1950s and you do the math until now it's a lot of years.
Yeah, it's been there since 1950s. And if you look at it, 5 years ago, we were in the verge of World War II. Am I right?
5 years before 1950s?
1945.
Oh yeah.
Yep. Correct me if I'm wrong, ladies and gentlemen.
I mean, I don't know. I just like, yeah. Okay.
Okay. So knowing that this extensive history is like happening for this piece of equipment, which is very helpful for today's lab condition and environment. So maybe you can tell us more, like you can give a more in-depth explanation of what a microplate reader is all about.
Yeah, sure, I will. Anyway, having this podcast in the lab really like boosting up the mood of like a scientist slash researcher slash technical support in me.
Yeah, you tell them, sister. Okay.
Okay, so anyway, so what was the question again? Like, what is microplate reader, isn't it? Okay.
No, but it's like the in-depth explanation of a microplate reader.
Okay,
Yeah.
enough explanation of. Sorry, I'm a bit distracted. Might have ADHD for sure. Okay, so the history, Mr. Shami here already explained it. It started in 1950s, but then what is the application? What is the microplate reader itself, isn't it?
Yeah.
Okay, so microplate reader itself it's involving in the study of light. But then when we talk about study of light, why does it related to biology or something?
Yeah.
Isn't it?
Why?
So because like when you talk about light, you might be thinking of Big Bang Theory of Sheldon's and the gang,
Oh,
right?
I don't-
But then it's involving study of light to detect the biology thing.
okay.
All right, so to detect the concentration of solution, to detect the viability of cells. So to easily to say, it is actually invented by all the physics stuff to be used in biology setting.
So it originates from the physics department.
Definitely all the engineer people are from physics.
Oh.
Anyway, so it is built by an engineer for sure. All right, and then used in biology setting. All right, so how does it use? So technically when we talk about microplate reader, it is very, how to say, it is the sound is not very complicated. It is not complicated actually if you know what you are doing with it. But to talk about it, microplate reader have a lot of variety. It's not only consisting of one. Like we can see here, like behind me, beside me here, this is like the most basic one, so it's very simple. So technically in microplate reader, we have 3 types of detection. The common detection, which is absorbance,
Okay.
fluorescence, and luminescence. So when we talk about absorbance, as the name is it is, right? Absorbance. So technically it's absorb
Okay.
light. So when we talk about absorbed light, so how it measured concentration of any solution or any tissue or anything, viability of anything. So it's because the detector in microplate reader itself is detecting the amount of the light that has been absorbed
I see. Okay.
by the solution. I mean, when you talk about absorbance, so you just imagine that you have a Coke in a clear glass. All right.
When you say Coke, you mean the drink, right?
Yes.
Okay.
Okay. Like the soda, the cola,
Okay.
right? Okay, so you just imagine that you have that in a clear glass, and then you have a torchlight.
Okay.
And then you put it beside the glass, and whatever that passes through is very little from whatever that it's it's started from, which is your light source.
I see.
So, which means in that case, some amount of light already been absorbed by the solution.
Yep.
Right? So that some amount of light that has been absorbed is what we call it as absorbance.
Okay. Yeah.
So
That's pretty straightforward.
yeah, so that is one of it. And secondly, it's fluorescence. All right, fluorescence technically, you measure the light that fluoresces from the sample.
Fluoresce. So it's like a plankton.
Yeah, you can see that.
Okay, I see.
So for fluorescence, lightsaber. So it's also fluorescence, isn't it? Yeah, it is fluorescence. It is a concept of fluorescence.
Okay.
I mean, come on. So it's like you can see it obviously it is fluoresce from there. So technically for fluorescence, it is actually needed some sort of enzyme inside it to actually get excited, and the energy, like how it get excited, the enzyme. Basically it's from power source, from the light source.
From the light source.
So that is why we said it's study of light. Right, so you need to expose the enzyme with the light, and it got excited, and then the energy will eventually go down. It will be emitted. So when it emitted, then that is when you measure the fluorescent signal.
I feel that that's a really good explanation for all the non-science people out there.
Yeah, so like for example, right? So when you talk about fluorescence, so the light that has been used or that is still being used in fluorescent detection is all the common fluorescence that you can see in lightsaber itself. Yellow, green, red. Yeah, so that is like the basic of the fluorescence color. And it is fluorescent like that. It don't just like light up like absorbance. It's just only using one source of light. No. So it's using like that kind of
So many lights, yeah.
yes, it's like a many layer of
Many
light.
layers of lights, yeah.
Exactly. And then the third one, we have luminescence. So luminescence, it's easy when we talk about jellyfish or firefly.
Oh, okay.
Right. So that one we call bioluminescence because it come from the organism
The
itself.
body
So
itself, yeah.
yes, so bioluminescence is like in living cells.
Mm-hmm.
So we actually, like scientists back then when they invented luminescence, so they take all the chemical inside of this firefly and even jellyfish actually to familiarize it outside so that people can repeat it back inside the lab.
I see.
So the formulation of the luminescence and everything, so it is originated from that.
I see. I mean, it caught my attention that fluorescence is like it reminds me, I know we have lightsabers in the fiction part, but we also have so-called lightsabers when it comes to the local tom yum shops.
Exactly.
Yeah.
I want to say that, but I want to be a bit professional, so I said lightsaber.
Yeah, but all these colorful, fancy lights that you see in all the local shops, actually, when you look at it the second time, you feel like, oh, you learned something because you actually have this idea
Exactly.
after listening to this podcast. So now it's very simple and understandable that all these three applications are all different on their own. Yeah.
So actually, I want to add more. So like how they're actually detecting from how to determine how much light, how much absorbance of fluorescent, or whatever. It based on the wavelength. Right, so you need to instruct the machine on certain wavelength. So each wavelength actually represent different colors.
It's like a Pink Floyd album,
Yeah.
Dark Side of the Moon.
Yeah, so each wavelength, so if you like the Pink Floyd, so they have like the prism
They
thing,
have a prism,
right?
yeah.
So it's like a lot of rainbows. So each of the color actually represented by different wavelength. So maybe for 60 nanometer, which is the unit for wavelength, it might represent the blue or green or something like that. So I mean, I couldn't remember well in each of the wavelength, but that is how it is like how
Differentiated.
how to differentiate for each wavelength represent different colors. So each application that people do in science, they should know at which wavelength they should actually use
So are
to
you saying
detect
like
that.
for example, like in the rainbow, like common normal occurrence in the environment, those are all like represented by different wavelengths also.
Definitely, because it's refraction of light.
Awesome.
It just we don't know. Like, can ask God.
Wow. God is the master of physics. Awesome. Okay. Okay. So we've covered all the applications, and I believe that most of the things that we hear today or just now is actually very like inspiring, and there's a lot of new things that we can learn from there. Yeah. So Zara, as a technical person, what maybe you have encountered so far, like in labs, or the labs that you have visited, they might feel a bit overwhelmed when it comes to like having to come up with a new microplate reader, uh because they're chasing with technology. So do you think, or do you know what are the challenges that they have to face so far? Because as I know it, all the microplate readers have these same applications, but it depends on like what kind of application they have in one machine, right? Yeah.
True.
Yeah.
Usually the overwhelming coming from the beginners.
Mm.
No matter in research, no matter in diagnostic, all right. So usually they are overwhelmed by this because they don't know like how do they want to actually use which wavelength or what kind of detection.
Mm.
Even me back then when I was doing my PhD, then I was like, oh, what do I do? Which one should I choose? So don't worry about that because usually, not usually, like in this era, in these years, so people don't really do things conventionally,
Okay, so they do
which
it
me
unorthodoxly.
not orthodox, also not conventional. So they actually currently it's more systematic.
Oh, it's so systematic.
Yes, definitely not orthodox. All right, so it's more systematic because usually when you are doing things, for example, so when we talk about microplate reader, I bet some of our listeners, some of them might have experience in using microplate reader.
Yeah.
So yeah, so basically, if you're talking about microplate reader, you will be very familiar of it's easy to usually when you talk about microplate reader, it's very familiar with protein work, for
Yes.
example,
Yes. Of
right?
course. Yeah.
Okay, so the overwhelming and everything, and they'll be like, okay, at what for protein, at which wavelength you need to. So first, what you need to do is, of course, you need to do a literature review.
Yes.
So you need to read a lot of reading. I mean, if you even do a quick Google search, it help people. So please.
Mhm.
So one is doing that, and second is actually when you buy kits or when you buy reagents, it's stated in their product sheet. It's stated in your technical sheet. You can refer to that. And then thirdly, if you cannot do all that, or you be like, oh, I don't have internet, and second, I couldn't download the product sheet or the
In the
product.
forest. Yeah.
Exactly. If you stay on the tree, I mean, I'm not offending anyone,
Yeah.
please.
Of course.
Yeah.
Yeah.
Okay. So
Maybe
I'm
you were
just
just out hiking. We don't know.
so metaphorically speaking, it that way. So if you don't have any access to all that,
Mhm.
but if you can access on SMS or WhatsApp,
Yep.
you can actually go to the company that where you purchase your kit and ask them. So at which wavelength? Because it's normal. So the overwhelmed part is more to they don't know
Yes, they
at
don't know.
which wavelength. Or sometimes for those who are beginners in research, for example, right? So for example, we talk here, we put cell culture people. So they wanted to know whether their cells, like what is the percentage of their living cell of or the dead cell. Yeah, so that one also you need to use microplate reader, to actually know the percentage of your for the cells that are still alive or dead. So yeah, so microplate reader is there. So that is why you need to actually do some sort of literature reading.
Yep.
And from there you can find. But believe me, as someone that has experience in research and also in industry,
Okay.
product insert really help you. You just need to actually find in box or anything or online. You just find the kit that you're using. For example, the viability cell, the percentage of viability cells is not that I mentioned, right?
Okay.
So you just do a quick Google search. Percentage of viability cell assay wavelength.
Insert as in a pamphlet, right?
Yes, insert
Okay.
mean the pamphlet.
Yeah, that's why you don't throw out your inserts, everyone. Just
Yeah.
yeah, it all comes for a reason. It was in there included as a reason. Yeah.
I have a suggestion.
Yeah.
File your insert. Kit insert.
Wow.
File it. It will be useful for you and your successor in the future.
But the thing is, if you try to file an insert, because all inserts come in different shapes and sizes,
True.
so we can't exactly like we can exactly expect everything to be the same size, right? So it's very hard for us to yeah.
Scan it and save it in a file, Google Drive. I mean, we're living in 2024.
Yeah, that's one thing that maybe all the manufacturers can take note. Yeah. Yeah, the phone can also scan. Yeah, unless if you accidentally delete it. But we're way off topic right now.
Yeah, you need to. I easily get.
I know, me too. I got distracted. Yeah, but okay. Actually, the question I asked just now means a lot to me because I was also a student doing their research back then. I was one of the students that did this research that uses a microplate reader, but this reader at that time I don't know what's the application and what is it all about, but all I know is I have to use it. But then again, we know that the limitation of universities, they don't really want to like invest in something that is better. And what I had was the basic one. I didn't know that all these applications existed and why the university didn't want to like I don't know, invest in a better microplate reader. Yeah. So having to know all these challenges, it helps me to think that there are two sides in every side lah.
Definitely.
Yeah, maybe as a person, maybe if I work in a university, I would know for sure these challenges and why we couldn't give the best for students. Yeah.
True enough.
Speaking very realistically.
Yeah, realistically speaking, that is the situation.
Yep. But as a student, I often wonder, what are the common myths that people actually like encounter when using the microplate reader, because sometimes I heard a lot of things saying, hey, you can measure a lot of things inside there. You can measure honey, for example. Like, what?
Actually, I think can, as long as but then again, you can measure, but then what is the purpose that you're measuring?
Yes, the
Exactly.
purpose.
So of course you can measure, then you can get the absorbance reading. For example, if you use absorbance, which is like the basic light, you can use it for absorbance reading. But for what? Again,
Yeah, there's no purpose for that, right?
yeah, I mean, there is some formula that you can use to measure concentration and everything, but then it's not easy as it is, because for you to identify concentration, you need to have your standard. What I mean by standard, you have to know known concentration of a series of, for example, honey dilution.
Okay.
So, for example, you have like 5 types of honey, and you know each of them, what is the concentration. Right from there, when you know the type of concentration, you run it in your microplate, in your microwave plate.
Okay.
And then after that, only you insert your wild honey or whatever
Ah,
type of honey. From there, you can plot the graph and find the concentration.
I see.
Yes, you cannot just simply like have the absorbance. Okay, I minus this with a blank on it or nothing. That one you can just get an absorbance reading, but for what purpose?
Okay, so it actually depends on the research itself.
Exactly.
Okay. So having said that, we know that the technology it's a trouble to like keep up with all the technology that is happening nowadays, right? So what do you think future, well, in a way, this microplate reader that we have in stock is actually like well equipped with the most current technology, right? I would say that.
But yeah, that one is like, what do I think about that?
Okay. What do you think
But
about that?
before what do I think, you asked me about the myth earlier.
Yeah.
I haven't answered that yet.
Oh, I thought the honey part. Okay, you actually asked.
Yeah, I'm answering your question about the honey. Can you measure honey?
Okay.
But the myth that people don't know, that people always say that you don't put a microplate reader beside centrifuge.
Oh, that's a myth? Like, oh, okay, I didn't know that.
Yeah. I mean, that is a myth for certain model.
Huh.
Why
Why
do
is
I
it?
say that? For a certain model, it can become a myth. Okay, technically it's a fact. But then in the old technology, they have a mirror inside their microplate reader.
Yes,
Right?
yes.
So there is filter, and
Okay.
inside there they have a light source, and they have mirror to reflect the light and everything.
Okay.
The mechanism inside. Right? So the mirror is easily to misalign. So when you put beside or like same bench as a centrifuge, the centrifuge will definitely will vibrate vigorously and everything, especially when you want it to slow down. It
Yes.
will cause vibration and everything. So it will affect the mirror, the mechanical mirror inside the microplate reader. So it will cause misalignment.
Ah,
But it become a myth when we have these days monochromator inside the microplate reader to replace the mirror, right? So what is monochromator?
yeah.
Monochromator is actually a mirror light device, but actually it's not a mirror at all. And then they have like a grafting gear shift that keep on rotating based on the user instruction.
I'm trying to imagine it right now.
Okay, so you just imagine it's like a concave thing.
Okay.
All right, so concave is like mirror-like, but it's actually not mirror, but it's just a device.
So it
So
was that
it's like a upgrade mirror, I guess.
replacing the mirror, yeah.
So it's like a device. That's why I say it's a mirror-like device.
Uh-huh.
But
Okay.
then we call it monochromator, and it has a grafting gear behind it to actually control the movement. So for example, when we talk about wavelength, so let's say you insert 420 nanometer. So the grafting gear behind the monochromator will like that, like will move the thing. So when you put it on the same bench as centrifuge,
Okay.
it will not affect because it is very robust.
I see.
So it will not get misaligned as easily as mirror.
Yeah.
So that is why I said on certain model, that fact become a myth.
Okay. I don't know whether you guys have heard of this myth before. Maybe in the research researchers feel it is something that is well known. I'm not a researcher.
I mean, yeah, there is like, oh, you don't put microplate reader beside centrifuge. It will affect your reading, everything. Yeah, people get scared of it. But as the technology evolve, the mechanical inside also evolve parallel as the technology.
Technology.
So they replace all the sensitive part. I mean, not all model, but the one that we carry thermoscientific have that.
Yeah, it's not a product placement, everyone. But yeah, we're trying to say that the micropredator that we have right now is actually well equipped.
Yeah, well equipped.
It's well equipped.
Yeah.
That's all I can say. But yeah, not even a product placement. But okay, I would say that's the most like future proof thing that we can add. Maybe
Yeah.
the provider has.
Because I always do talk from institution to institution.
Okay.
So this is kind of talk that I try to create the awareness, and I would say that's all this while I would say that I have been like about 10 institutes in this year itself, like in 6 months. So I've been like to more than 10 institutes to give talk about microplate reader,
Wow.
and none of them know about that, about the mirror that has been replaced by monochromator, like total monochromator, because certain model they have monochromator, but they still retain the mirror structure inside the machine.
I see.
But
Now
then
everyone knows now.
exactly. So certain model they already replaced totally, total mirrors no more.
Amazing. I mean, yeah, I wouldn't know until like a few seconds ago.
Yeah, good
You
for
mentioned
you.
this. Okay. Knowing that, right? Of course, there are a lot of different opinions or different feedbacks that you can get from these customers that you see and all these lab people, right? So, what are like the challenges of you highlighting these particular features to these people?
Usually for because when we talk about research field, right? So to highlight about new technology, it's easy to tell students about it because they are aware. Even a phone, every year, sometimes it's even 2 models come out in a year. So they are very open to a new technology. They can easily believe that, oh yes, the technology really have advanced rapidly. But when we talk about microplate reader to older generations, or maybe like the older researchers, they are a little bit, what is the word? They are a little bit
Obnoxious? No, that's a very harsh word.
no.
No, they're a bit skeptical.
Yeah, very skeptical. They are very skeptical. They'll be like, can they believe for the new technology? I think old technology is more stable.
Okay.
While actually old technology require them a lot of money
Effort. Yeah.
effort, money to actually maintain, to actually use, and it's not even variety.
Yes. Yes. True. It's a waste of time.
Exactly.
Why don't you want to?
So that is the biggest challenge for me as someone who actually trying to give awareness.
This applies to everything actually in the world today because there are a lot of new things happening almost every day, but it's always a challenge to always like change the old mindset.
Exactly.
Yeah.
And even I have personal experience when I did my study. So the older I would say supervisors or the older generation researchers, they'll be like, oh, you can use the older one, it's more stable. While us, we want result fast and we want it to be accurate and we want it to have reproducibility.
Yes.
So of course we go like for us like younger generation, then we go to the better options that we have.
Yeah, of course.
Yeah.
It can help, right?
Yeah, exactly. It can help. I mean, it's called new for a reason, right? So ended up for us like younger generation, we all going to the of course a better option, a faster options rather than we use the
The old
Venetian
school.
one.
Yeah.
I would say.
Well, yeah, I don't know whether this is gonna be like in your face thing to other people, but yes, I've heard this in one sitcom that I always watch. New is always better. Sometimes.
Sometimes.
Sometimes. Okay.
Not applicable for all situations, I guess.
I think we are running out of time, but maybe I'll run with you like only one question. If you can explain a microplate reader in one keyword to a non-lab person or non-scientist, what would that be?
One keyword.
What's one keyword? Just one word.
If one, can I go 3 words?
Okay, if you want to. I mean, one is like the most minimum thing.
Microplate reader is not. It's not only a reader. I mean, it might be 6 words already, but yeah, people. So microplate reader is not only a reader. You can do a lot of stuff in it, like the software in it, the application in it. You can actually explore, try yourself, explore it, especially the software. Each of microplate reader have their own software, so just explore the software and
I don't know. I might want to say it's like a computer of some sort. A computer that measures liquid.
I mean, yes, but then I know that the time is running out, but
Yeah.
I wanted to add a little bit
Go
more.
ahead. No worry. No
Okay,
worry.
so because like we are talking about microplate reader, it's not only a reader, right?
Okay.
So the main situation now is people are just using microplate reader, taking the absorbance reading,
Yes.
and go home and process it in their Excel.
Okay.
But a lot of people don't know the software that actually equipped with the microplate reader can do a lot more than that. You can do your analysis directly from there.
Okay.
All right. So from there, you don't need to do like in ancient way, which is you go to Excel, and then you plot one by one, and then like you plot the graph and find the concentration. No,
So troublesome, man.
I mean, back then it's not troublesome because I know that people receive the template from their seniors.
Which also received the template from their previous.
Exactly, and then it's you don't know like how good how true is the template because you
Yes.
will never check. You will just say, oh yeah, my seniors use it, so I will use it again. So so by doing that, it didn't actually making you like a smart person, I guess.
Yeah, because you're just following the old tradition, which might not be
It's
true.
a routine, mundane thing that you'll be like, oh yeah, we follow the previous one. So by having the technology evolve, you actually can explore a lot more.
Yep.
A lot more using the software itself. So everything can be done. You don't even need a calculator.
Just a little bit of PSA for everyone's listening. I would say that we really need to change the landscape of scientific.
Exactly.
Yes, the landscape of science in Malaysia right now, because we have this culture of always following the tradition, of always following the things that was done by our previous what do you call it, supervisors. It's like we really need to do something about it, because if we don't change, we cannot leap forward.
Exactly.
We cannot always rely on the old ways. It's always better.
Exactly. I mean, we are the younger generation need to break the chain.
Yes. So yeah, it's not like you are just learning about microplate readers today. Well, it is more than just a reader. You learn more life lessons now. Okay. So yeah, I think that's about time. That's time.
Okay.
Okay. Thank you everyone for listening, and thank you so much, Dr. Zara, for being with me today to discuss on this one heck of a machine.
Yeah, if you let me talk, I will talk continuously. I will not stop, but luckily you stopped me.
Good to know that. Okay, I'm also grateful that I can stop myself from talking. Okay, anyway, if you want to hear more about Dr. Zara talking, well, you can always join our FCBIOS webinar series. We will hold like maybe 1 month. That's 2 times or 3 times depending. Dr. Zara might always be the speaker, or she might not join. Well, it depends, but you can always find her there.
Busy, guys.
Yeah, she's busy. But anyway, we can also have our live recording session, and every recording of the webinar session in our on-demand webinar platform, which is in the FCBIOS website, www.fcbios.com.my, and it's in the resources section of the website. So yeah, head over there and find out if you want to.
You can join my tech talk too if you want me to talk more.
Yeah, Dr. Zara can actually go to your places to give a talk, like a personalized talk on webinar. But maybe if you want to invite her for another podcast, be my guest. I mean, but she's busy lah. You need to ask her.
Need to book my time.
You need to book her time. Yeah, she's basically booked for the whole of December, guys. So Okay, so yep, I guess that's a wrap up.
Right, thank you so much.
Thanks, Dara.
You're welcome.
And we'll see you next on the next podcast series, FC Bios podcast series brought to you by FC Bios. Bye bye.